This procedure demonstrates how to measure the strength of intracellular cohesion in 3D tissue-like aggregates. The first step is to generate spherical tissue-like aggregates and to transfer these into a tissue culture medium-filled petri dish. Next, individual aggregates are aspirated into a Pasteur pipette and loaded into the lower compression plate of the tensiometer chamber. Then, the upper compression plate is connected to a nickel-chromium wire, which connects it to the electrobalance and establishes a zero baseline weight referred to as F0. The experiment is initiated by raising the lower compression plate until the aggregate becomes compressed between the lower and upper compression plates, and is terminated when aggregates reach shape and force equilibrium, or FEQ. The final steps of the procedure are to capture images, measure aggregate geometry and force of resistance, and to input these measurements into the Young-Laplace equation. Ultimately, results can be obtained that show changes in tissue cohesion and how these can influence tumor dispersal or spatial positioning between different cell populations through aggregate spreading assays or cell sorting assays respectively.
So this method can help answer key questions in the fields of tumor biology, embryonic development, and tissue engineering. Questions such as how does tumor cohesion influence malignant invasion, the stratification of germ layers during gastrulation, or how can we actually engineer cohesion to generate functional organ-like structures in vitro?
Experimental manipulations involving the culture of living cells should be performed under aseptic conditions in a designated laminar flow hood.